Hello, hello. diversifying the न चोरहार्यं न च राजहार्यं न भ्रातृभाज्यं न च भारकारि व्यये कृते वर्धते एव नित्यं विद्याधनं सर्वधनप्रधानम्. Neither the thieves can steal, nor the kings can confiscate. Can it not be divided among brothers, and will it have no weight? To spend it as much as to earn it even more. To say that the wealth of knowledge is superior to all. To be here has reminded me of what Dr. Theodore Sezesel said. The more that you read, the more things you will know. The more that you learn, the more places you will go. A warm good morning to all the distinguished dignitaries, esteemed speakers, faculty members, and all the participants present here. It is both my honor and privilege to welcome you all to this prestigious event organized by Research Center of Genetics and Genomics with Genotypic Genomic Simplified of Centurion University of Technology and Management, Bhubaneswar. I, Divya Jyoti Das, esteemed member of Research Center of Genetics and Genomics, am delighted to serve as your anchor for this national conference on 20th and 21st February 2025, Frontiers in Genetics and Genomics. Centurion University, established in 2005, began by revitalizing Jagannath Institute of Technology and Management in Odisha. In 2010, it became Centurion University of Technology and Management, Odisha's first multi-sector state-private university. With the mission shaping lives and empowering communities, Centurion University of Technology and Management focuses on grassroots development through education, training, financial services, and community projects, uplifting rural communities in Odisha and Andhra Pradesh. Centurion University Center for Genetics and Genomics, pioneering advancement in biotechnology, agriculture, and healthcare. Through student-centric project-based learning, it cultivates future leaders. Experimental learning in cutting-edge areas like genome editing and speed breeding, combined with a design-driven pedagogy, foster innovation and prepares graduates for dynamic field. The National Conference on Frontiers in Genetics and Genomics 2025, sponsored by ANRF and co-organized by Genotypic Genomics Simplified, will convene experts to distinguish cutting-edge advances in genetics and genomics, focusing on gene editing, sequencing, personalized medicine, and computational genomics. The conference will foster collaboration and explore future research directions to improve human health. To officially commence our conference, I would like to invite our esteemed dignitaries to the dais. Please join me in welcoming Professor Supriya Patnaik, Vice Chancellor, Centurion University of Technology and Management, Professor Arjula Ramachandran Reddy, former Professor, University of Hyderabad, and founder Vice Chancellor, Yogi Veranna University, Professor B.P. Acharya Sir, retired IAS, Special Chief Secretary, Telangana, and Dr. Rukmini Mishra, HOD, HOD Botany, School of Applied Science, and Centurion University of Technology and Management, and coordinator of Research Center of Genetics and Genomics. Now I would like to request the volunteers to offer token of sapling to the guests, a small token of ours, a symbol for growth and new beginnings. Shubham karoti kalyanam, marogyandana sampada, shatru buddhi vinashaya, deepam jyotir namastute. The grantor of auspicious welfare, health, wealth, and prosperity. Salutation to you, O flame of the lamp, for the destruction of the thoughts of enmity. And now, to symbolize the illumination of knowledge and the igniting of our shared passion, I invite our esteemed guest to kindly proceed to the lamp lighting ceremony. This traditional gesture represents the dispelling of darkness and the beginning of a bright and insightful conference.
शुभं कुरु त्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरु त्वं कल्याणम् आरोग्यं धनसम्पदः शत्रुबुद्धिविनाशाय दीपज्योतिर्नमोऽस्तु ते दीपज्योतिर्नमोऽस्तु ते दीपज्योतिर्परब्रह्म दीपज्योतिर्जनार्दनः दीपज्योतिर्परब्रह्म दीपज्योतिर्जनार्दनः दीपो हरतु मे पापम् सन्ध्यादीपनमोऽस्तु ते ओ दीपब्रह्मरूपस्त्वं ज्योतिषां प्रभुरव्ययः ओ दीपब्रह्मरूपस्त्वं ज्योतिषां प्रभुरव्ययः आरोग्यं देहि पुत्रांश्च मतिं स्वच्छां हि मे सदा रवेरस्तं समारभ्य यावत् सूर्योदयो भवेत् रवेरस्तं समारभ्य यावत् सूर्योदयो भवेत् यस्य तिष्ठे गृहे दीपः तस्य नास्ति दरिद्रता शुभं कुरु त्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरु त्वं कल्याणम् आरोग्यं धनसम्पदः शत्रुबुद्धिविनाशाय दीपज्योतिर्नमोऽस्तु ते दीपज्योतिर्नमोऽस्तु ते शुभं कुरु त्वं कल्याणम् आरोग्यं धनसम्पदः
Now, I would like to request everybody to be stand at their respective places for the national anthem. Moving into the today's event, we have Dr. Rukmini Mishra, who is currently serving as an associate professor and head at the Department of Botany, School of Applied Sciences, Centurion University of Technology and Management, Bhubaneswar. She also serves as the group leader of the Research Center, Center of Genetics and Genomics. Ma'am has a PhD in Agriculture Biotechnology from National Rice Research Institute, Cuttack. Ma'am is the recipient of the prestigious Young Scientist Award from the Department of Science and Technology, DST, Government of India, and the prestigious Talented Young Scientist Fellowship under the TYSP program of the Ministry of Science and Technology, Government of China. Recently, Ma'am has received the prestigious CERB International Research Experience Fellowship from the DST Government of India to work on advanced genome editing and technologies at the University of Missouri, Columbia, USA. She teaches graduate and postgraduate courses on plant biotechnology and plant genomics. Ma'am have published over more than 40 research articles in journals of national and international repute. Currently, ma'am is running a DST-CERB Government of India funded project towards utilization of gene editing technologies for the disease resistance in vegetable crops, and also a project on endangered medicinal plants funded by NMPB, National Medicinal Plant Board, Ministry of Ayush, Government of India. Now, I would like to request ma'am to kindly grace the gathering with her insightful words. Ma'am.
Thank you so much, Divya ma'am. A very good morning to one and all present here. Honorable Vice Chancellor Madam, Professor Supriya Patnaik, esteemed Chief Guest, Professor Arjula Ramachandra Reddy, Guest of Honor, Professor B.P. Acharya, sir, esteemed dignitaries of the dais, distinguished speakers, faculty members, students, participants, ladies and gentlemen. I extend a warm welcome to each one of you present in the hall to this 2 days national conference on Frontiers of Genetics and Genomics, which is sponsored by Anusandhan Research National Research Foundation, ANRF, Government of India, and it is also co-sponsored, co-organized by our industry partner, Genotypic Technology Private Limited, Bangalore. So this is the first national conference which is organized by our research center, Center for Genetics and Genomics, and I'm very happy that we got the support from ANRF for this. And also I'm equally happy that we have co-organized this with our industry partner, which we are collaborating from last 3 to 4 years with this company, and they have been a great support for us. So thank you, team. So before, in last 3-4 months, we have started preparing this, that we'll be organizing a national conference. And while we were having discussion, we thought that instead of making it on a very broad area, we'll make it very specific, because our Center of Genetics and Genomics, we are working in these few areas, like genomics-assisted breeding, gene editing, informatics, genome engineering, gene sequencing. And so we felt that we must have the best of minds, the brilliant speakers from industry, academia, the upcoming startups and research institutes, so that we can have a very good discussion. We can have the insightful discussions, who can discuss the groundbreaking discoveries, and if there is any problem, the scientific problems, how the industry and academia collaboration can come together, and people from different institutes can come together and see how we can solve these problems, or if there is any issue, and what exactly the advancements are, how should we keep in touch with them, and what should we do to see what advancement is going on, and what is it lacking, and how we can solve it. So this conference will provide a platform for you all. Mostly the participants are students, so I'm very happy that they will be highly benefited. They will get an opportunity to discuss with people who are from industry, and they can see how the world is, what exactly we are doing, and what the market needs. So it will be a very great platform. So I wouldn't waste much of the time. I wish you all the best, and I hope that you will have a great session. Thank you.
On behalf of all the attendees, I would like to express my sincere gratitude to Dr. Rukmini Mishra ma'am for outlining the importance of the conference. Moving ahead, it is my distinct pleasure to introduce you all to Professor Supriya Patnaik ma'am, who is our Vice Chancellor. Supriya ma'am has her qualification from the Tata Institute of Social Sciences, MA, and National Institute of Mental Health and Neurosciences, India, and RMIT University, Australia. She has extensive teaching and research policy experience, and her research interest is in the field of gender and development, poverty and social development, sustainable livelihood, skill-integrated higher education, research methodologies, and social work pedagogies in different contexts. Ma'am has several publications to her credit. Ma'am has also worked with NGOs, multilateral and bilateral agencies, federal and state government, and universities in India and Australia. Ma'am is currently serving as Vice Chancellor of the Centurion University of Technology and Management, Odisha, India. She is an industry licensed person with RMIT University, Melbourne, Australia, and also research fellow at the Private Limited Global Competency Assessment and Accreditation Glides Private Limited. To our few names, she is also on the board of governance of the Chetna's Institute of Management and Research, Mumbai, India. I would like to welcome her to address the gathering with some insightful words. Ma'am.
Good morning. Welcome one and all. Professor Arjula Ramachandra Reddy, Professor B.P. Acharya, my colleague Dr. Rupa, guests for the tour who here to attend the conference and be part of it, my colleagues, participants, and dear students. It is indeed an honor to welcome you all to the National Conference on Frontiers in Genetics and Genomics 2025. Step forward in bringing together bright minds to discuss groundbreaking advancements in genetics, biotechnology, and allied fields. At Centurion University of Technology and Management, just to brief you a little bit for the benefit of the outsiders, that we are a private university who is designated as a skills university, and we have 6 campuses in very remote geographies, and it has been an excellent, what should I say, it has been the work of a team of people to bring Centurion to what it is today. I think you all are aware that as part of our skills university, we also have several enterprises that have been promoted by the university, which are also into commercial production of various things. Like we also have a transformer production unit, we have e-vehicles production unit, we also have apparel production unit, and so on. This is just a very brief about it, but at Centurion University of Technology and Management, we are deeply committed to research and innovation. Our Center for Genetics and Genomics is at the forefront of genome sequencing, gene editing, and molecular marker development. We focus on precision breeding, disease resistance, and sustainable agriculture solutions by integrating advanced sequencing techniques and CRISPR-based editing. We aim to tackle real-world challenges in crop science and biotechnology. This conference is an opportunity to exchange ideas, build collaborations, and contribute to the future of life sciences. I encourage all of you to engage in meaningful discussions, learn from experts, and explore new research directions. I extend my heartfelt thanks to the organizers, speakers, and participants for making this event possible. Your contributions will shape the future of genetics and genomics. Wishing you all a successful and insightful conference. Thank you.
Thank you, ma'am, for your insightful and inspiring address. Your words have provided a valuable framework for today's event. I'm honored to introduce our guest of honor. Today we are delighted to have Professor B.P. Acharya, sir. Sir is a retired IS officer, former Special Chief Secretary of Telangana. Sir holds a key position and launched innovative initiatives. He developed Genome Valley, felicitated numerous industrial clusters as MD, APIIC, and established district knowledge and innovation centers. Post-retirement, Sir advised on industrial cluster development, served on government committees, and is a professor of practice at Centurion University of Technology and Management. Now, I would like to request Sir to kindly grace the gathering with his words. Sir.
Professor Supriya Patnaik ji, Dr. Arjula Reddy garu, my good friend, Dr. Supriya Patnaik, Rukmini Mishra. Because you're all over the place, I get confused. And the distinguished faculty who are attending here, delegates of this important conference, and young students, the students who are attending. We especially look forward to the students to carry on with the innovation which Madam mentioned just now. It's been, my association with Centurion has been a very interesting journey since last 1 year. Last time when I came here, I said I'm a freshly minted professor. I worked as a civil servant, but they made me a professor of practice. So I take great pleasure in coming back and interacting with the students and the faculty. I will start with a small story, how genetics and genomics needs to be given the importance it deserves. It was 2001 or 2. We went to China, visited Beijing Genome Institute. There we saw a poster that BGI has decoded the rice genome. But I was aware the Professor Arjula Reddy's team was already working on it and they had decoded. We had decoded much before China did, but they were claiming the credit for decoding rice genome. Human genome was decoded little earlier. So I came back and told the Chief Minister, here is a group of scientists working on the rice genome, and they have decoded. So immediately said, call them, we will get it presented. Dr. Kota Harinarana, in Odisha, he belongs to Parlakemandi. He was the Vice Chancellor, and Professor Arjula Reddy was the professor in the life sciences. His student, Dr. Markandeya, who is coming tomorrow, he was also there. I have that photograph I'll share with Rukmini. They came and they presented the rice genome sequence CD. Those days CDs were popular. CDs to the Chief Minister. We highlight cDNA clone CD, yeah. CD, DNA cloning CD. So that is how genetics and the decoding of genome was given importance in Hyderabad University under Dr. Reddy's leadership. He has trained nearly 30 years of genetic students in Hyderabad University and in very important committees in biotechnology related committees. But unfortunately, R&D still remains on a back seat in India. We still take pride in reverse engineering, making up generic drugs, which is the easier way of doing it. This new fund which is created, Anusandhan National Research Foundation, hopefully it will spur the research which is much needed. I was again very happy yesterday to see a facility of Bharat Biotech in the outskirts of Bhubaneswar, hardly 15-20 kilometers from here at Andharwa, Odisha Biotech Park. They have invested 1,500 crores by making a state-of-the-art vaccine manufacturing unit. It will produce 8.2 billion doses of vaccine per annum, including the malaria vaccine, which is the first in the world. Countries abroad, the countries in the West, big pharma is not interested in vaccines. They want to make drugs. They want to make costly drugs, cancer drug, a diabetes drug. They want to make money. It is the countries like India, the developing world, we have to take the lead. India has the capacity, has the science, where we can provide the answers. That is where the challenge lies. That is where the academia industry interface is very important. What Centurion is doing now is the right thing, and I hope the genetics department, under the leadership of Rukmini ji, goes ahead and sets up the bio-incubator. The ecosystem of science must come up here. Students must get involved in doing research, not only getting degrees. BTech biotechnology is no use. In fact, many of our students are working in Bharat Biotech. It's called Sapigen. They are subsidiary of Bharat Biotech, Sapigen. They already have about 100 students working, including this lady who was serving coffee. She's also from Centurion. She is trained in your cafe. I was surprised to see. So you were spread into multiple skills, including catering, including the food service, the F&B sector, everything. So the professors, they are aware. In fact, I invited them to come and attend the conference here today. There's another link with malaria and Hyderabad. Maybe many of you don't know. The first Nobel laureate from India came from Hyderabad, Ronald Rose. He found malaria parasite in Hyderabad in 1820. The company from Hyderabad, malaria vaccine, first time in the world in Bhubaneswar. So that is the link between Hyderabad and Bhubaneswar. So we provide this bridge. So I welcome this, the bridge of knowledge, this nexus of good as they call it, and future. Apart from the bio incubator that we are planning in our university here, also in Parlakamandi, Agri-Biotech Incubator. Both these will come up soon. We hope to create startups which will tomorrow add to the ecosystem. I requested Bharat Biotech to allot land to small startups around that. That's how Genome Valley came up. Dr. Krishna Ella came in 1996. We started around 2000 and created a cluster of 5,000 acres, which is the largest biotech cluster in the world today, life science cluster in the world. No such, many cities tried, they never succeeded. But important is to create the right ecosystem, right kind of environment so that the industry grows instead of creating hurdles for the industry. That is where the challenge lies. That is where the bureaucracy, the civil service needs to be proactive. The politicians must have the vision to create it. People like Ajula Reddy, who has tremendous knowledge base, they need to be given a free hand in creating new ideas, in sharing ideas. Conferences like this will go a very long way in creating this exchange of ideas and seeing and inspiring the students who will be the R&D experts for future. I hope and pray that the biotechnology, genetics, and the genomics department here grows fast and creates the right kind of interface between academia and industry. Thank you very much for inviting me. It's always a pleasure to come back and meet you all. Thank you.
Thank you, sir, for your enlightening words. Now it is my honor to introduce our chief guest for today's gathering, Professor Arjula Ramchandra Reddy, sir. Sir is an emeritus professor at the University of Hyderabad and a fellow of the Indian Academy of Sciences. A distinguished scientist, educationist, and academic administrator. Sir has served 2 terms as Vice Chancellor of Yogi Vemanna University, establishing 27 departments and significantly expanding faculty within a short period. Sir's expertise lies in rice genetics, genomics, and biotechnology. With a focus on drought tolerance, Professor Reddy's research conducted in his world-class laboratory has garnered international recognition and funding from the prestigious organizations like the Rockefeller Foundation and the European Union. Sir has served on numerous national and international committees related to agriculture, biotechnology, and GM crop regulation, including co-chairing the Genetic Engineering Appraisal Committee of India. Sir's contribution to science and education have earned him numerous awards and honor. Now, I would like to request Sir to kindly share few words with the gathering. Sir.
Good morning to all. At the outset, let me thank the authorities of this university, Centurion University, and Mrs. Rukmini, Dr. Rukmini, and other organizers for giving me this opportunity to be here. When I first saw this university, some thoughts came out to me. But first, Here, for 45 years or so, simply remained as a Sarkari professor. Here is my neighbor's Sarkari IAS officer. Now he shifted to be professorship, but I didn't shift to that. Okay. Now, Centurion University, in my thinking, rightly came in right time at right place. Here is a situation where there is a gap that exists between theory and practice. And that gap, we at the national level were trying to fill up that gap through many academic committees, academic and administrative committees, and industrial committees, but never realized, because there is no structured university for this purpose. Here is a private university taking risk to fill that gap is really amazing, because we have experience by sitting in so many committees. This is the real uniqueness of this institution, in my opinion. Fighting to fill the bridge between theory and practice. That's one thing. And it's doing the good job. Supriya ji just by a slip of a tongue said molecular marketing instead of molecular marker. That is exactly what is needed. Molecules have to be marketed. These molecules have to be marketed. Otherwise the molecules will remain in the lab, in the test tubes. Right. So therefore, all kinds of molecules have to be marketed. Now, you must be surprised to know, India was one of the earliest country in the world which started a bureaucratic structure for a framework for biotech spread. That was rules 1986. The rules 1980, it's called euphemistically the rules 1979. And then 1986, then 1990, from that onwards, I was in those committees for 30 years. So we know how I am a direct spectator or participants of the biotech growth in this country. Everywhere, we spend a lot of money every year, but practicality is something still we have to see. What was happening was, a lot of projects we gave, I was a part of the committee, so we gave chairman of the committees and all that. We gave many projects to plant geneticists, but plant molecular biologists, but sometimes we somehow missed breeders, those who make really things happen. Somebody has to breed a good variety to eat, for us to eat. That gap was missing. Publications were coming on plant molecular biology, biotechnology, genomics, sequencing, and all that, which is in those days very romantic. But later we realized those molecules are not getting into the plant, therefore not getting into the market. So now situation is different. Given the bureaucracy and I don't use the words, but the kind of troubles I went through in pushing BT brinjal and BT cotton hybrids, I took a lot of risk in clearing 1,800 hybrids of BT cotton. 1,800 hybrids. That pushed the production of cotton in India like anything. But I have to take a lot of nonsense from so many people, so many organizations. There are a lot of people always to obstruct the progress. Very few people to encourage the progress. In democratic country like this, that's what they call by mistake freedom of speech. Okay? But freedom to obstruct science is not a freedom of speech. It is not. It's the other way around. Anyway, so I'm happy to be here to share our work and our thoughts about this university and about the overall ecosystem that you people have generated. And I see a lot of gender parity is not much here, because not so bad here. So many women I see, scientists here. Actually, they dominate the number here. Looks like that. But sometimes in biology, as a vice chancellor, I realized every biology class I go, there is already domination. The number is in favor of women students. That's a good sign. And I think a lot more research can be done without disturbance. There are a lot of girls. So it's good for administration, it's good for the students, because I went through this. So I wish you all, now all you have to do today, the students, the delegates, is just see what kind of benefit you get out of these talks. Primary goal. If you don't do that, if you just listen and delete it, like a computer thing, just delete, and then go home, then in next Janma, incarnation, you will be born as crows, you keep talking, not listening. So, so therefore, it's the best thing not to be born as a crows. You please take much interest in this subject and ask questions, because I believe no question is a stupid question. So don't hesitate to ask. No question is a stupid question. Not to ask a question is a stupidity. Okay, thank you very much.
I would like to express my sincere gratitude to Professor Arjula sir for his insightful and inspiring words. A collection of thoughts is a form of self-portrait, summarizing the very essence of the event we have gathered here for. We have National Conference on Frontiers in Genetics and Genomics 2025 Book of Abstracts. This book of abstracts isn't just a collection of summaries. It's a testament to the vibrant and dynamic community we have here for. It's a tool for collaboration, inspiration, and the exchange of the knowledge that is so vital to the scientific progress. Now, I would like to request all the dignitaries to officially unveil this invaluable resource, National Conference on Frontier in Genomics and Genetics 2025 Book of Abstracts. Now I would like to request Professor Supriya Patnaik, Ma'am, Vice Chancellor, Centurion University of Technology and Management, to kindly felicitate our guest of honor, Professor B.P. Acharya sir, and Chief Guest, Professor Arjula Ramchandra Reddy sir. Now I would like to request the dignitaries to kindly occupy the seats. I would like to request the dignitaries to kindly occupy the seats downstairs. Now, I would like to request Professor Arjula Ramachandra Reddy sir to kindly address the gathering on the topic plant genome evolution, movers and shakers. Sir.
which on about the plant genomes. This is a very, by default, this is very complicated hardcore genetics, but I tried to simplify as much as possible because I am aiming this talk to students who are not a 100% geneticists, they are molecular biologists, biochemists, biologists and so on. So, I have simplified this deliberately to understand, to make it clear and clear so that you can take some information into your textbooks. Okay. Now, plant genome evolution is a very complex process. In fact, all evolutions are. Evolution of animals, bacteria, everything is complicated, but plants give you a much more complicated structure. And what are the factors that move and rearrange and affect the evolution significantly? This is what the title of the talk is. So, this is how my talk will be organized. So, think that I am giving a classroom lecture actually, not a technical, deep technology session. This is the organization of my talk. First of all, plant genomes are complex. They display unparalleled variation in size, unparalleled variation, not present in any other kingdom, not animal kingdom at all. The plant genomes are highly dynamic, they are not static, highly dynamic. Non-plant DNA is a major constituent of plant genomes. It's interesting. Plant have a bona fide plant DNA, but extra plant DNA makes major constituent of those genomes. Not in humans so much, but plants, and I will tell you what are those unwanted guests in the plant genome which is pushing up the evolution. Something is moving plants. These are called the transposable elements called mobile genes. As you students of genetics, you know, today if the gene on a human chromosome is present, on the 6th chromosome it is present, after 300 years also it will be on the same 6th chromosome. But we have been working on this gene, and first I will tell you the history of that. But these genes have a capacity to move, to jump around. Today they are on 6th chromosome, tomorrow they may be on the 9th chromosome. While jumping, it will disturb the genome. These are called genome disruptors. And this is a peculiar characteristic of some plant DNAs. So, I will give you the structure, function, and evolution of transposons. Transposable elements are mobile genes, genes that move, unusual. And how the Ts expand the genome. The genomes are expanded and contracted, unlike human genome. Not much difference, but in plants they do. And how do they proliferate? How the plant control them not to burst, not to colonize the human genome or a plant genome. How Ts regulate plant gene expression and evolve new traits, altogether new traits, altogether new. That means a new gene is created. Normally genes evolve through mutations, variations, but new genes are created by these mobile genes. And are they major movers and shakers of plant genome more than a plant genome itself? Yes. That's why we ask the question, how human is a human? How much human is a human actually? Okay. So, the serial genome case study, I will take better example, that's where we worked, so we can give you the best example that's available today. Okay, here we start with, look at the genome sizes, the 1C value, so-called 1C value, haploid genome content, okay, total DNA in the haploid system. You can see now flowering plants, where is this? Could you see this? No. Here, yeah. So, if you see plants, it starts from 10 to the power of 5 kilobases, that is into 1000. From here all the way up to here, you see other animals, algae, fungi, nowhere near. Only amphibians are close to this, about a billion nucleotides, 4 billion nucleotides or so. Now, this is funny. Why plants should have so much DNA? Flowering plants have the largest genome on planet Earth. Many people were not aware of that, but there is a reason why they should have it. So, if you say, second thing you notice here, they vary size from here all the way to there. So, by a factor of 2000. 2000-fold DNA increase in the plant kingdom, flowering plants. That attracted attention of a lot of people, and it was hard, but some of us could look into that actually, what's wrong with this and why it is happening. So, flowering plants, summaries of this talk is right now is flowering plants have the largest genome on planet Earth, and the size variation in this among the genera, in the plant kingdom is huge. Humans genome don't vary much. We are also 0.01, but look at that, variation is from million base pairs to 150 gigabases. This lowest plant, smallest genome plant, smallest genome is only 67 megabases. That's million base pairs. But the largest is 1 gigabase. That's megabase, that's gigabase. Okay. You see now, plant genomes do not stay put. If they stay put, just static, there won't be such a size variation. Something is happening in the plant. And contract, and these sequences are diversified. Plant genomes, now I'm not talking about plant evolution. Plant genomes are evolving through rearrangements, through a lot of churning around going on in the plant genome, fueled by these elements. And what is more interesting is, the smallest plant, Genelesia, is 61 megabases. That's 61 million base pairs. But Panicum japonicum, the Panicum japonica, is 150 gigabases. 150 gigabases. The little plant here, the little plant there, and the very big plant here, they have same flowering, reproduction, seed, leaf, photosynthesis, photorespiration. All the functions are there. If the smallest genome plant and the largest genome plant have the same functional complexity, why so much DNA? What was the need for that DNA? Why was it there? And how in the first place it came there? That's an interesting question evolutionarily. That's what I try to decipher for you today. Okay. Now, okay, but genome is expanding. Gene number is not increasing. This is another thing. So non-genetic DNA is increasing, genetic DNA remain more or less constant. The little plant genome, like Arabidopsis, is about 27,000 genes. The biggest genome, like one of the largest genome is like wheat genome, same number of genes. So what is the rest of it? What is that DNA which is not genes? Okay. You know, by this time, all DNA present in a cell is not a genetic DNA. They are not genes. Okay, only a fraction of it. Okay, so now let's see. Among the plants, there may be some botanists here. Among the angiosperms, you see angiosperm genome varies from here to all the way there. This is where Panicum japonica. Now, this the green one is the so-called gymnosperms. Gymnosperms have larger genomes than the angiosperms, flowering plants. Stupid, you know, they don't flower, but they have more DNA. But the variation is for angiosperms from here to all the way there. Gymnosperms, all of them are big genome, but right here. So genome size variation in non-flowering plants, not typical reproducing same reproductive machinery plants. They have less small genomes. They have big genome, but variation is small. This variation, the reason for variation is the subject of this today. Okay. Now, so the question comes, if that is not more genes, what is the DNA then? What is the 90% of DNA? 80% of DNA is not plant DNA in the plant. What is it? So this is a classification done long time ago. Now you see here, plant genome contains unique DNA sequences. This is not more than 1 to 3% maximum. Rest of it is all repeat DNAs. Satellite DNA, microsatellites, mini satellites, so much of DNA, so-called junk DNA, what you call, but they are not junk. But the problem is, there are three types of repeats. There are tandem repeats, which is ATGC, 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 or non-tandem, ATGC, reverse tandem, CGTA, so on. But more important is this dispersed repeats. They contain a lot of DNA that encode retro-pseudogenes, retrovirus gene, and retrotransposons is viral, DNA transposons, signs, lines, I will tell you what they are. So bulk of the DNA is in the repeat DNA. People classified that as junk DNA. It's not junk. It contains the movers and shakers of the plant genome. That's what stunned the public, the scientists at the time, world over. Okay, so now we are concentrating only on this side of the story. This side of the story, transposons and DNA transposons, long terminal repeat retrotransposons, signs, lines, mites, and so on. I will show you what they are. Okay. So here, now, how much? When you say that plant genomes vary, there is a lot of species across the plant kingdom. You pick up so many species starting from maize here on the top. This is the intensity of transposable elements. If a genome maize genome contains 80% of transposable elements, 80% of maize genome is not maize genome, not maize genes. Only 20% is a maize genome. Like that across, more than 70 species have been fully sequenced. And then search for, you can see there are some which have 80%, sorry. There are some which are 80%, maize is 80%, and then there are some other thing you can't read it, this. It's a lot of work, but you see anywhere from 20 to 80% are transposable element, that is, they are distributed across the plant kingdom, no exceptions, and they present in a diverse positions and quantity. That's what it means. Now here you see the genome sizes. The maize genome size is about 2,500 megabases, 2.5 gigabase. It's a bigger serial, bigger diploid serial. Now you could see there 80% of that. Then barley, then so many, and there are some which don't even have Ts, very little Ts. And here other species where genome sizes are represented. The genome size big, the transposable element size also big. So as the genomes increase, Ts also increase. So the net summary, indirect conclusion is It is a transposable element that increases the size. That is the reason for variation in the genome size in plants, starting from less than 65 or 70 megabases to gigabases. One of the reason is the proliferation of this DNA. That's proven across species. Whenever the genome size increases, you see the transposable element size increases. So one is causing the other. Okay. Now, what is more important is, as a student of botany, you know, plants develop, plant genomes develop, called polyploidy. A diploid becomes tetraploid, like triploid grapes, no seeds. 3 genomes. Okay. Potato, tetraploids. Wheat, hexaploid, 6 times. Allohexaploid. There are autohexaploids also. So genome doubles, 1 to 2, 2 to 4, 4 to 6, so on. That is one reason for the expansion of the genome. That's what the large genome, that's why they are large. But that's not the only reason. That's not the only reason. Sometimes 2 diploid species where there is no chromosome doubling, they differ in size. How? That means there is something other than polyploidy which is pushing the size, increasing the size of the plant genome, making fatty. Some plants have obese plant genomes. Some plants are on diet. They don't go for proliferation of a lot of DNA. So this slide shows very interesting fact. These are all rices, different kind of species. That means they're in one genus Oryza. One genus Oryza, 2 varieties of Glaberrima granula. Some of these rice varieties we took it from Katak, which is called Makkah of rice, okay? Makkah for rice research, rice germplasm resources. Now you see here Oryza granulata, 2 varieties of rice, and there is a difference. You see, these are 2 species of rice. They are different size. They are diploid. There is no chromosome doubling, but they increase the size. That is purely because of the elements, jumping genes, what you call. Story is same, Glaberrima nivara, which is the progenitor of rice, sativa, again differ in sizes. Some of them small, some of them are small DNA, Brachyantha is small, but again, Australiensis is a huge rice genome. They're all diploids, no chromosomal doubling in this. That was the first clue that one could get, is that polyploidy is not the only reason for genome expansion. This is the non-plant genetic material within the plant is kicking this action and making DNA bulge, what we call. Okay, so there is a spectacular size variation among the plant kingdom. Some diploids have a larger genome than their tetraploids. Chromosome doubled 2 times. Diploid, haploid to diploid, diploid to tetraploid. Yet, non-tetraploids are larger than tetraploid within the species, within the genus. So that means something is happening. Morphological variation does not match with genome variation. Theory of evolution, what do you call? Evolution by variation and natural selection, not genome selection, not genome evolution. So the morphological basis of evolutionary criteria is different from the genome evolution criteria because of this problem, because we are dealing with a non-plant genetic material. Those are not subject to natural selection because they have nothing to do with reproductive fitness. 1 minus S, so-called reproductive fitness is equal to 1 minus S. That is not feasible here. That doesn't apply here. Okay, so now you see, summary, take-home lesson for you is that plant genome size spectacularly varies across species, genera, and across families, Gramineae family, order, Poales, and so on. They vary within the family, within order, within species. So 3 levels. And most of the DNA in plant chromosome does not belong to plant itself. This is a drastic conclusion. One of the most abundant gene families in plant genomes encode reverse transcriptase-based RNA sequences. So, what are these? That will be the next part of the talk. One of the reasons for genome size variation is abundance of long terminal repeats and long terminal repeat Ts, just LTRs and LTR-Ts. So we will tell you what those are. genome expanded, genome expands when LTRs become active, genome contracts when the plant tries to delete these sequences. Okay, polyploid is a major player, but not the only one for genome expansion, right? Okay, so what is the plant genome architecture looks like? Fundamental issue. Plant genome contains the total DNA, and that total DNA is organized into genes. Genes are organized into gene families, and gene families are organized into chromosomes, and chromosomes organized into genome. That's the progression. Now, if you increase the size here, if you increase the size at the gene level, you increase the genome size. That's what's happening here. Okay, so plant genome evolution appears to have gone through repeated bulging and shedding. That means plants get obese. Here, when you get obese, you go to a bariatric surgery. The plants also have a mechanism of getting rid of extra DNA. It won't allow transposons to keep on increasing the DNA size, which kills the plant anyway at the end. So that won't happen. Evolution has provided the plant machinery, molecular machinery, that will restrain the transposons and getting rid of them. Small, large, and whole genome duplications, deletions, repeat sequences, retrotransposons are responsible for increasing or decreasing the plant genome size. This is a summary of this, all those slides. Okay. Now, yeah, this is the sorghum, jowar on the left, and maize on the right. Sorghum genome, these two are separated by 7 million years. Separated in the evolution by 7 million years. The genome of sorghum is a little small, 700. This is 2500 megabases. That means about 3.5 times increased genome size, and both of them are diploids, no chromosomal doubling. So somehow these transposon elements preferred maize to keep on increasing the size, which sorghum did not get through. So what we call here maize has become very fatty and obese. Sorghum is on diet. That's why millets you eat. Okay, so they have a meaning. Genome size difference is 3 times, greater than 3 times bulged, same speed. I mean, they're all cereals, they're all grasses. All you eat, maize, wheat, rice, barley, all those are grasses. Originally grasses, which you see everywhere in the lawn and everywhere. Man interfered with them, selection, sequence, selection, breeding, and finally we have a nice product to eat. Okay. So now, so there are 2 types of mobile elements. The DNA elements, that is a DNA to DNA to DNA. They are in the plant, they replicate, they become transposable elements. But there are other kinds of elements, RNA viruses, sequences, and these are from DNA to RNA. Then RNA is reverse transcribed into DNA, and that enters into the plant DNA. It's a very nice system. Here the virus is so clever, it will make its own RNA unique, and that RNA is reverse transcribed. Normally DNA to RNA to protein, that is a molecular dogma. But here RNA to DNA, and the DNA gets into the chromosome. So it doesn't allow the host plant to just kill the RNA, it transcribes by itself. So the RNA transcribing DNA is in the chromosomes. It goes as if it is a host DNA. It escapes attention of the DNases. Foreign DNAs normally are chopped off by DNases in plant, but not this. Because this is reverse transcribed DNA, and that gets into the plant chromosome, just like that. So that is the problem. So later, first discovered in maize by Barbara McClintock. Barbara McClintock was a great scientist, the greatest geneticist ever lived on this planet Earth. And she discovered this in maize first when DNA was not known as genetic material. She discovered there are some genes that move. Nobody knew what the gene was. Some people thought genes are proteins. At that time, she discovered this. But she has to wait until she becomes 80 years or so, then she got Nobel Prize. Discovery was 1940s, late 1940s. Okay, I'll tell you some nostalgic about this. Yeah, this element is the one, this structure of mobile gene, cartoon of that, not a whole structure, discovered by McClintock, not molecularly. She knew by genetic analysis some gene was moving from one chromosome into the other. And she explained how it is able to move, which is 100% true even today, after all the molecular biology done. Now, about 50 years ago, this month, February 50, 1975, I was awarded PhD for working on a gene in which this was present, but I didn't know that. And same month, I went to USA to finally work on this itself. That's why today is very nostalgic to me. That's why I agreed to come actually here, because that reminds me what we have done 50 years ago, which most of you are not born. Okay? Yeah, this is a 75. I went to Iowa State University where McClintock's graduate student. Then he became professor. He brought this seed material, and that attracted my attention because what I was working in university, I knew there was an element, it was misbehaving it. It was moving from place to place, but there was no way, no DNA is known, nothing is there at that time. So I knew. So therefore, luckily I wrote to him and he took me up. And that guy with whose lab I worked is a PhD student of Barbara McClintock. So I also had a 10 days work with Barbara McClintock. That was enough. She was so genius, so brilliant. It's impossible to keep up. That is why she has no PhD student. She had no PhD student and she got Nobel Prize for all that work done. Very tough to work with her. Okay, so this element has a unique structure. It's a bulk of the, it's a 4,650 nucleotides long, okay? And what you see here is the sequences, end sequences are very, very unique. End sequences, there are 5 nucleotides here which are flanking direct repeats. When this element sits into a chromosome from one place to another, it goes because of this. So these are called transposon footprints also. So when it leaves, it creates a duplication of 5 nucleotides. Some elements put 10 nucleotides. And there is an inverted terminal repeats. That is same sequence, one end of the element are like this. So you read that carefully. Now you see here, for example, here, ACGCT. See, now ACGT. Now ACGCT here, ACGCT. ACGCT, upside down, palindromic. You understood? This is a unique structure of this. A direct repeat and indirect terminal inverted repeat. If any sequence has this in the genome, what is there inside the genome, ORF, this produces an enzyme, very unique enzyme called, encodes unique enzyme called transposase, which will work as a DNase, also integrates both functions. It allows chromosome to be cut. It also allows chromosome to be pasted. Yeah. So it is autonomous element. This is simply it can sit in any place in the chromosome. If it sits into a normal gene in humans, let's say, this kind of elements exist in humans, then that gene becomes a mutant, end up in a disease. It can. Okay. Its jumps are more in somatic cells. They don't matter in the human genome. But if that happens in a germinal cell, it will you transmit to your children transmit to their children, it goes on. Okay. So this is whenever such ends exist, that's a site for scissors. It will cut. The genome is floating, the DNA is floating, it will enter into the chromosome. Very simple. Okay. Yeah. So the McClintock's element is called the AC element, activator element, SPM element, EN element. They're all does. This gene, this moving gene, has 5 exons, 4 introns, and a subterminal region and a terminal inverted repeat. These are the landmarks, hallmarks of a transposon. Now, if I get rid of this exon 1, 2, 3, or 3 and 4, or 1 and 2, the element becomes inactive. It will finally sit wherever it sits in the chromosome. Let's say it just established itself into the round and wrinkled gene, round gene of Mendelian gene, sweet pea. You must have studied round, wrinkled, red and yellow, yellow and green and all that. That will behave like many of the mutants that Mendel studied were actually transposon insertion. They were not real mutations. So if a transposon inserts into a gene and not moving out, if it's into ORF, a framework, it will create a framework mutation, and then that's the end of it. The gene is not functional. But nothing is wrong with the gene. If you somehow make this element jump out, the gene will become normal. Okay, that's constantly happening in the plant. I will show you. Okay. Yeah, see here, the McClintock's own thing. And I brought this seed back to India. This is actually from our lab. Now, maize 9th chromosome has 3 genes, color gene, bronze, and vaccine. This is 9th chromosome short arm of maize. Okay? There is a DS element, which is a transposon, same AC element, minus a portion of it. So minus central region. The AC element I showed you here. There, the DS element loses something here, some deletions here. Then it cannot jump. It requires the activity of this element to make that as a jump, to make that element jump. So you see now what's happening. So here, there is a chromosome, the DS element. This is a derivative of the AC element that breaks the chromosome. Now look at here. Centromere is here. These are 4 genes. There are 3 here because DS is not there. Now break. This area it's broken. Wherever DS is there, it's broken. So this CIBZ wax, the phenotype is colorless. This is colorless. This is this. Now, when this happens, you need acentric. You get into acentric fragment. There is no centromere. This is chewed up by the DNases. This is chewed up. That's chewed up, and the CBZ, this become colored now. From colorless, it will become colored. And mitosis occurs, then this is finally lost, and CBZ wax colored. So that happens in a developing seed. You see, basically start with colorless with this, and whenever there is a jump outside, the chromosome is broken, a red spot will come. The gene is restored from colorless to color. So number of colored dots on a cob tells the frequency of jumps. This is the assay for us all the time, those of us who worked with him. The assay for that is how many times it jumped out. And you see some variation in size of this. If a jump occurs very early, all the cells developing from that will be the same phenotype colored. But if a jump occurs last cell division, only small tiny dot is there. Only 2 cells will show. So transposons can be used as a developmental markers. And that is exactly what the jumps take place. Okay. Now, so these are the cobs which I brought from that lab to India when I joined as an associate professor at the University of Hyderabad in '78. So you see the variation of the colors. So you start with this. We target this color gene. This is a C gene on 9th chromosome short arm. You target transposon and to jump into this, and then cross into colorless, you will get this kind of segregation, F2s. So from colored to colorless, you can push it. Colorless to colored also, you can push it. And such 500 cobs we brought from India. At that time, bureaucracy was not so harsh, so they just allowed anything. So everything is allowed. So big container, and so 6 of our students worked on 6 of these colored genes. Okay, so we now have the whole pathway, and this pathway is a mobile. I mean, this whole pathway we transferred by cloning, sequencing, and transforming rice. All the maize genes were put into rice. Same colors in rice too. Okay, now, okay, so this is the McClintock element I told you, and if all these internal portions are deleted, that becomes DS1. DS is a derivative of AC. It's a derivative of AC. So this is a filler DNA. This is spacer DNA, unknown, not coding for anything. Similarly, there is a DS element with 2 exons, so partial, truncated proteins, truncated, no protein at all. So obviously, there are a lot of churning going on within this element, and when it's deleted, this thing cannot jump. None of these can jump. Only that this element makes them jump. So all I need to do is take a normal maize, cross it with a line containing activator, then you see the jumping around going on. That's how we trap it. And we score only that trap element, whichever gene we want. Okay? So I don't want to go into details of these complex mechanisms. That will put you to sleep. But here what it looks like, cartoonist. Here we see the normal chromosome. There is no AC here on the top. There is a DS element here. There is a normal gene here. Now, you could see the result. There is nothing. Everything is similar, same. This is a mutant. This is a wild type. Now, AC present, it breaks this. The DS is broken. Now moved proximal to the W locus. W locus. The result, it looks like this. And then it jumps out again, then it restored to normal. So it plays around like this. Insertion, mutation phenotype. Reversion, normal wild type. Oscillation. We can identify the frequencies of that. Okay, now, oh, sorry, we jumped. So look at this third one. Here, this one, AC. So these jumped into a mutation, into W locus. It's a mutation here. Now AC, this came back again. It made to jump back and then normally restored. So it wild type to mutant, mutant to wild type. So a lot of reversion mutants you see in nature are because of this. Okay. So these are the DNA elements. All maize transposon elements I described so far are DNA elements. Now this is the culprit. This is the RNA viruses, RNA transposons that make bulk of the plant DNA. That's I showed you 75% of the DNA. That is this. These are two types, copia and gypsy elements. So copia elements is a moving machine by itself. It's the primary binding site there, GAG protein, then the protease, then integrase, then reverse transcriptase, RNase H, and ORF, unknown ORF, envelope ORF. Okay, so they have this is a copia element, this is gypsy elements. Plant is made up of 60% of gypsy elements, irrespective of maize, sorghum, you just take among the transposons, 60% is gypsy. Normally 40% is copia elements. So these are the sequences, and they have one thing is common. But look, what is the difference between this? These are the same. It's bigger size, but you see the RT gene is here, int is here. Okay, so now int was here, it came to here now. Inside itself, it is moving around. That makes all the difference. Integrase, int is integrase. RNase H is an enzyme that detects RNA, DNA hybrids and chops it off. Okay, these are non-LTR retrotransposons. These elements will sit in the genome forever, and they make bulk of it. They are inactive, sitting in a gene, making it look like a mutant. You classify them as mutants. You classify them by, when you teach the students, you call mutants, mutants, mutants. Those are all this. Most of them are this. Okay, yeah. Now, this is the structure of these proliferating LTR elements. They have a long terminal repeat of 300 nucleotides. This is a target site duplication site. This is a GAG protein or reverse transcriptase, RNase H, int, untranslated region, LTRs, again, primer binding site. They are exactly same, more or less. Inside, these are the inactive RNA viruses, retrotransposons. So these are active retrotransposons. So both ways, it will activate in the gel, in the plant genome. Now, quickly, I will take as a case study to make you believe. You may be thinking that it is all theory. Yeah, 5 minutes. Yeah. So this is a rice study. All these are grasses. All these are grasses, but they differ in genome sizes. Okay. So here you see this. You started with the original rice, cereal genotype or the line that has gone through whole genome duplication once 70 million years ago. It became with 2 nested chromosome fusions. It became poidy. This is brachypodium, wheat, and rice on one arm, and the other arm is millet, sorghum, maize on the other side. But look, the genome size is here from this 2n, n equal to 12, here n equal to 2n equal to 20 here, and this is 2n equal to 42. Chromosomes number is increased, and transposons also increased in this. That is why this is a very good study for transposable element study. The genome differences here is rice is smaller, sorghum a little big, maize is very big, barley is 2, triticum yeast is 8,000 megabases, wheat is ultimate, 15 gigabases. Okay, so content of the transposons. Barley is 88% of the barley genome is not barley genes. 80% of it is not barley. Okay, so that is astonishing. Okay, now here you see copia element here and gypsy element there. Improved maize, that's what you eat today. Your maize, whatever you are eating, that's improved. 18,079 families of copia elements are there in this. 18,000 of them in millions of copies you have them. And gypsy elements, 20,000 of them. But overall, you have a common 1,10,000 copies of the transposons sitting in rice genome nicely, whatever we are eating today. Okay. And now you see the genome density on the top. This is gene density. Wherever genes are low, transposons are more. You see this? This is a transposon density. Blue. Gene density low, transposons are more. Gene density high, transposons are little low. So it goes around simply saying this requires years of analysis and data. Okay. Yeah, here you see the comparison from Arabidopsis, simple Arabidopsis model plant, and by the time you come to barley, how the genome is replaced nicely. The red is tDNA, normal plant DNA is blue. Now you see here, as you go by, you see the tremendous genome size not because of plant genome, the other genomes. Okay, this is for classroom. You can use this study. Here it is a transposon jumping into this and creating this kind of structure, a mutant. And there is one more here. It is sitting 2 copies on the other side, one on the other side, same direction. But you get a ring chromosome. Ring chromosomes are eaten away by the plant DNAs. So the genome size is low. This is how it controls. So these are all various kinds of the sequence rearrangements in plants. So transposons rearrange the plant genes, not only themselves, but plant genomes. And here, these last 2 slides, very interesting slides, maize work. Here is the maize chromosome number 1, alcohol dehydrogenase locus, normal gene. Here you see this element retrotransposon jumped into here. Sinful jump, we call sinful because it was one postdoc worked for 10 years to get this. So we call sin to work with it. So sin, but CIN is in Mexico. So the milt, fourth victim, Huck is a postdoctoral fellow in the lab of Jeff, where I also worked. So there are so many elements are there, we lost the names. So whoever working, graduate student, his name is there in this, short name of this. Okay, there is one on my name too somewhere. So you have an OP element, we have a grande element which is sitting. So what is happening here is one element sat in this, here. Some other element sat in that element. 3 elements sat in that. 2 elements sat in that. So piggybacking, one element sitting, one DNA sitting, another. In the process, what happens? If something sits, a little bigger. 3, 4, 5, 10 elements, much bigger. Chromosome is enlarged. This is what is happening. Okay. Now here, McClintock study. McClintock's 9th chromosome short arm here. And you see the busy haplotypes. You see how the genes are added. Now this blue sites, you see, it's not only genes it's adding methylation sites. So gene silencing is happening here. So epigenetic control they are doing. Okay, these elements are doing epigenetic control. And grande 1, for example, is in every maize element we looked at, it has grande 1. That's why it's called grande. Okay, they are TK, xeons, cacti elements, so on, so on, so on. They're full of them. Now this is the last slide where we show, here we take a small portion of the chromosome, 10,000 nucleotide sequenced of the same regions in all serials. Same region in all serials. This is called comparative genomic analysis. So you could see here there are 3 genes, red one genes, yellow is unknown genes, some kind of a junk sequence or spacer sequence. By the time you come to brachypodium, which is the originator, which is one of the simplest genome of cereals, it increased the unknown sequence. It also increased one transposon jumped in, and another transposon, blue one is jumped here. The blue one is LTR element jumped in. When you come to sorghum, 1, 2, 3 DNA element, and 4 and big. So if this chromosome small, it became big. When the question of rice comes, same area, same genes. Now you see one big transposon, one DNA transposon, one DNA transposon, one all, and the genes remain intact, so on and so on. By the time maize it comes, you see how many genes were added. How this chromosome has become big from here to here. So many things happen. So I didn't describe you what is long interspersed nuclear element, short interspersed nuclear element, miniaturized transcribing transposable element sequences and so on. All I tried to show you is chromosome size increases, DNA size, genome size increases just not because of only polyploid, but these non-plant DNA, which is retrotransposons, which are originated from retroviruses, and that increases the size of DNA. And meanwhile, they rearrange the plant genome. You see the variation. A lot of mutations it induce, they go as spontaneous mutations, for which you have no reason to, no explanation for them. And these can be oscillated in mutation out reversion. So this has implications in human disease. implications in plant disease resistance, plant abiotic stress resistance, and so many other factors. That's what amazes me. So we took this long to work on that for 30, 40 years. And what you call phonetics, transposon phonetics. We have some of us are called as a transposon phonetics, but it has implications in biology. I didn't show you more slides because already time is over. I tried to explain like a classroom lecture. Otherwise, it would have been completed in 20 minutes. Yeah, thank you very much. Any questions from any young student? It's going, but I tried to make it very simple. It's not that simple what I showed here, okay? Yeah, all right, you keep thinking. Yeah, yeah, please. Yeah, yeah.
Under certain stress in terms of surviving. Is there any?
You mean genomic stress?
Yeah.
Okay. So transposon size will, number of transposable sequences increase, density of the transposon increase when you make a wild cross between a wild variety and a improved variety. You see the drama, the dance of transposons we call in the F1s, hybrids itself. Okay. So genome has a, as I told you, genomes have a beautiful mechanisms. This is by three phenomena. One is non-homologous recombination. Another is abnormal recombination, a so-called oblique recombination, and by that it will eliminate. Okay, a transposon goes like this, and when it tandem duplicates, it's like this. But if it can also jump like this, then in between whatever sequence is there, it got deleted. So plant genomes have a mechanism to get rid of the transposons. Okay, but there are two types. So I told you one of them is a cut and paste. Another is copy and paste. It remains where it is. It copies. That copy moves into. That's class 2. So we have various means. The plant doesn't allow it to completely go bereft. Otherwise, genome size will, you know, if that goes, plant won't look like a plant then. So therefore, it has a mechanism to cut down because non-homologous recombination and aberrant crossing overs will get rid of this. It depends upon the orientation of the insertion. Okay.
Thank you, sir. We extend our sincere gratitude for captivating keynote address. Your expertise in plant genome evolution was evident, and your clear explanation of complex concepts made the topic accessible to all of us. It was indeed a valuable framework for our further discussion and research in this field. Thank you once again, sir, for sharing your valuable knowledge with us. We are here to add what we can to life, not to get what we can from it, by William Osler. With this, we move towards the conclusion of the inaugural session. Now, I would like to request our co-convener, Mr. M.D. Rahman sir, Manager, Genomics Product Unit, Genotypic Technology Private Limited, to offer vote of thanks. Sir.
A very good afternoon, everyone. It is my privilege extend a heartfelt vote on behalf of the organization committee. Thank you very much, Dr. Rukmini. And first of all, I want to thank you, sir, that the insightful of the presentation, it was very, very, very nice. Thank you very much for that. And first and foremost, our deepest gratitude, Professor Reddy, Professor Supriya, Professor Acharya, and Dr. Rukmini. Thank you to all our speakers from the different organization, different private organization, to who is going to give us, who is going to provide all the information, research information to us. Thank you. Thank you very much to all the speakers. A sincere thank you to all the sponsors, mainly for the genotypic technology, who is constantly supporting for this, as well as who is contributing in the CUTM with the research oriented. Thank you very much to genotypic technology. And thank you to all the students, all the faculties, all the supportive staff, each and everyone. Thank you very much for making this wonderful conference. Thanks a lot.
Thank you, sir. The conference will now pause for a tea break. I request all the dignitaries to proceed towards the hall number 5 for the tea, and request the participants to proceed towards the registration desk area. We will resume the conference with technical session 1 by 12:15. Thank you.